Snap-8, the Acetyl Octapeptide-3 protocol.
Hello, this is Peptide Proof, rigorous, independent, and evidence-based intelligence for the peptide science community. We track peptide breakthroughs from benchtop to bedside. Today, let's dive into the science of Snap-8, also known as Acetyl Octapeptide-3, and exactly how it modulates expression lines.
Let's start by looking right at the neuromuscular junction. Expression lines, at their core, are a mechanical failure caused by repetitive biological signaling. Every time we smile or frown, vesicles in our nerve endings release acetylcholine. Now, the critical bottleneck here, the structural vulnerability, is the snare complex. As you can see, it consists of SNAP-25, syntaxin, and snaptobrevin. You can think of it as a molecular docking clamp. Without this clamp functioning properly, that acetylcholine simply cannot be released.
So how do we intervene? There are two fundamentally different strategies. Looking at the destruction pathway on the left, botulinum toxin, commonly known as Botox, cleaves the SNAP-25 peptide bond entirely, resulting in total muscle paralysis. But competitive modulation, shown on the right, takes a completely different route. Snap-8 is a topical peptide that doesn't destroy anything. Instead, it reversibly mimics the N-terminal region of the native SNAP-25. The outcome is signal attenuation. This means softer, modulated expressions, completely without paralysis.
To understand exactly how this attenuation works, let's quickly walk through the 4 steps of the Snap-8 interception protocol. Step 1 is mimicry. This 8 amino acid sequence uses key residues, specifically glutamate, methionine, and glutamine, to precisely align with the natural binding interface. Step 2 is competition, where the peptide physically outcompetes the native SNAP-25 fragment. That leaves us right into Step 3, destabilization. The snare complex still forms, but it's functionally weaker. Finally, we reach attenuation. Vesicle fusion efficiency drops, acetylcholine payloads decrease, and over time, the mechanical force of those wrinkle-forming muscle contractions is significantly lowered. However, we do have a primary physics problem to address here, the stratum corneum logistics deficit. Notice the 500 Dalton permeability threshold on this chart. Passive diffusion generally fails for molecules heavier than this limit. Snap-8 weighs in at roughly 900 Daltons, and it's a hydrophilic molecule, so without a dedicated delivery architecture, an aqueous peptide like this is simply repelled by the skin's lipid barrier and ends up just evaporating on the surface.
To successfully overcome that penetration barrier, clinical science offers specific solutions. Right now, the strongest evidence lies with microneedle arrays. For example, a 2024 Shin study published in the Annals of Dermatology showed that dissolving microneedles bypass the stratum corneum completely, achieving measurable wrinkle reduction in just 28 days. Similarly, a 2020 OPSAL study achieved a 25.8% reduction in fine lines over 12 weeks using a multi-targeted hyaluronic acid patch. For standard serums utilizing chemical enhancers, efficacy strictly requires high concentrations of 5 to 10% active formulations, and they must be actively paired with targeted penetration fluidizers like glycols or liposomes.
Now let's position Snap-8 within the broader landscape of neuromodulating peptides. Arginine is generally considered our baseline. It's a 6-chain competitor targeting the snare N-terminal. Snap-8 acts as the 8-chain upgrade, offering a higher theoretical binding affinity to that exact same target. In contrast, Synake operates differently. It acts as a direct antagonist right at the acetylcholine receptor level. And then Lufacil works even further upstream to physically reduce calcium influx. Each of these provides a highly specific biological mechanism for tackling the exact same mechanical problem.
What experienced formulators know is that the true potential is unlocked in combination, creating real biochemical synergy. Stacking these orthogonal mechanisms allows them to compound. From the top down, the Snap-8 intervention addresses the root mechanical cause by reversibly modulating motor neuron signaling to reduce muscle tension. And from the bottom up, the matricil intervention addresses the structural consequence by signaling fibroblasts to synthesize new collagen. Because they target completely different cell types across different tissue layers, they do not interfere with one another. They simply compound.
That synergy, however, relies entirely on strict formulation compliance, specifically regarding pH sensitivity. Peptides are highly sensitive proteins. Looking at protocol violations here, layering Snap-8 immediately over strong AHAs, glycolic acid toners, or L-ascorbic acid with a pH below 3.5 is a major error. Doing this causes total acid hydrolysis, actively degrading the molecular chain before it can even penetrate the skin. The approved protocol dictates maintaining formulations at a neutral pH. If you must combine them in the same routine, you need to enforce a strict 15-plus-minute gap, or ideally, deploy your peptides in an isolated evening routine.
As delivery architectures evolve and synergy protocols become more exact, it leaves us with an interesting question. Will precision peptide stacking eventually render traditional injectables obsolete? That's all for this explainer. This is Peptide Proof, rigorous, independent, and evidence-based. We'll continue following breakthroughs in peptide science, from benchtop research to bedside therapeutics, with the scrutiny they deserve.